And first of all, I would like to acknowledge Oxford Nanopore for selecting our work to present it here in this very interesting session. So to start, it is always helpful to give some data and some perspective of the problem that we are dealing with, which is rare diseases. Rare diseases, as we know, they are rare. They affect 1 in 10,000 people, but as there are over 6,000 different rare disorders, we are talking about a condition that affects around 4 to 6% of the global population. So it means that we are talking about 300 million people all over the world. And it is always important that the 70% of these people of rare disorders have a genetic base. But as we know, the problem is the diagnosis. In most of the cases, the time of diagnosis lasting 4 to 5 years. And also, despite the great development on technology, there's still up to 35% of patients with unresolved genetic base. And also, the 70% of these cases have a misdiagnosis. So there's a clear need to cover this diagnostic gap, and that's why it's important our research here. So with this introduction, the objective of our study was to use nanopore sequencing technology to trying to fill this gap. And for that, we have studied 4 different rare disorders with intermediate phenotypes and samples with a no molecular base as a proof of concept of the usefulness of this technology in rare diseases. And we have analyzed 2 different groups of rare disorders, 2 hemostatic disorders, one of them antithrombin deficiency, which is dominant and increases the risk of thrombosis. And in the other way of the balance of the hemostasis, Glanzmann thrombasthenia, which increase the risk of bleeding. And 2 metabolic disorders, peroxisomal disorder and glycogen storage disease. These last 3 disorders are recessive. And for that, we have conducted the samples were selected from large groups of samples recruited by 4 specialized reference centers in Spain, specialized in each pathology, in which they have done a complete molecular diagnosis algorithm following conventional studies like Sanger sequencing, whole exome, MLPS, NIP array, and unresolved samples were selected to this study by nanopore sequencing technologies. And you can see that we have 13 samples with antithrombin deficiency, 2 samples with Glanzmann thrombasthenia, and 1 of both peroxisomal disorder and glycogen storage disorder. So we have conducted this study using 2 different approaches. We have a study with the PromethION 12 samples, and as we have selected samples with candidate genes, we could perform nanopore sequencing in the MinION device using adaptive sampling of the target that we are going to study. So here I show you some metrics about the runs. As you can see, we have obtained a mean read length of 10,000 base pairs in both techniques, PromethION and the MinION. As you can see, the average coverage in obtaining PromethION was of 17x. That was non-uniform depending on the quality of the DNA of the samples studied, during a time sequencing of 72 hours. And in the cases of the MinION device using adaptive sampling, it is interesting that we obtain a 7x of coverage of single libraries running during 30 hours in our flow cells. And the data analysis was different between these 2 approaches. For the PromethION samples, a more complex multimodal pipeline was designed because these samples were included in a larger group of samples. So this pipeline included alignment with Minimap, structural variant calling by sniffles, and intra and intersample merge in order to search our variants. And a simpler and in-house pipeline was conducted in our lab for the MinION samples. It's a straightforward structural variant discovery that includes also alignment with Minimap, a structural variation calling by sniffles, and EGV visualization. So going through the results, From the group of samples, of 13 samples with antithrombin deficiency without any candidate variant, we have studied 12 samples in the PromethION device. And by this approach, we were very surprised that 3 samples were resolved. We found an insertion of a retrotransposon, SVA, of 2.4 kilobases that was inserted in intron 6 of serpinC1. This is the first insertion of a SVA element in a hemostatic disorder. As we have the exact sequence, we could perform a de novo assembly and characterize that it was antisense-oriented with a tandem site duplication, and we described that it was new SVA element close to the SVAE in the phylogenetic tree. And as we know the exact sequence of this element, we could validate it by PCR by designing an internal primer in the sequence inserted of the SVA and an external one in serpinC1, because as you can see in this image, it was impossible to amplify the whole element inserted by several primer design that we tried. So we validated by PCR in this case, in this sample, and the relatives, and also it supported the founder effect of this retrotransposon insertion in these unrelated cases. So the next sample resolved by nanopore sequencing was studied by, or sequenced by, in the MinION device, enriching the region of serpinC1 of 3 megabases of the genome in this region. By this strategy, we found another retrotransposon. In this case, it was inserted in intron 3. It was a 2.9 kilobases of a SVA element. And we performed the same strategy. By de novo assembly, we fully characterized this element, and it was a SVA-F1, which we studied it, and it was specific found in humans. We fully characterized it by bioinformatic tools, and we validated as in the same strategy as the previous one, with an internal primer and another in the serpinC1 region. So now we are trying to study the origin of this retrotransposon movement and insertion in our candidate gene. Going to the next disorder, Glanzmann thrombasthenia. As you remember, it was a recessive disorder. We have 2 unresolved samples. In this case, we have 1 sample with any mutation, and we have another sample with a pathogenic variant, a single nucleotide variant, in one of the 2 genes responsible of this disorder. So we performed the MinION sequencing, targeting in this case the 2 regions of the 2 genes of interest of this disorder. And by this strategy, nanopore sequencing detected a complex structural variant affecting ETGB3 gene involving exon 2 to 6. And it was very interesting because it was a deletion, inversion, and duplication that impairs the structure of this gene. So in the sample 1, it was, as you can see in this image, it was in homozygous state, confirming this candidate variant. And in the second one, it was in heterozygous state in the alternative allele of the pathogenic allele of the mutation already found previously. That was detected by nanopore, and we could phase the variant and demonstrate that it was in the alternative alleles. This is another strength of this technology that allows us the phasing of the variants. So again, we validate all the breakpoints by PCR, designing the exact primer to amplify the breakpoints. And we also validate the relative, so the segregation of this structural variant, and again, demonstrating the founder effect of this variant in these unrelated samples. The next disorder studied was glycogen storage disease. In this case, we had 1 sample with a pathogenic variant, a single nucleotide variant in GS2 gene, but again, it's a recessive disorder, so it was not sufficient for the diagnosis. So we studied this by nanopore sequencing in the MinION device, and we detect a line element of 1.5 kilobases in exon 10 of GS2 gene. And the final disorder was peroxisomal disease. We have this sample with a pathogenic variant, not enough for the diagnosis. So nanopore sequencing of the gene, the region of interest, result in an insertion, an insertion of a retrotransposon SVA in intron 8 of PEX2 gene. So all in all, in summary, we have seen that nanopore sequencing has filled the gap of the genetic test in 8 samples from 4 different rare disorders with a known molecular base by conventional methods. 4 in antithrombin deficiency and 2 and all the samples that we have selected samples from the other disorders. And what we found so interesting, and I think that also for you, is this very complex mechanism, the retrotransposon insertion that is hidden by other conventional methods that we have seen involved in 6 samples in 3 different disorders. So the conclusion of our study is that nanopore sequencing, including adaptive sampling of candidate genes using MinION device, is the most suitable method for the detection of structural variants underlying largely unresolved cases with different rare diseases. And retrotransposon insertion, which are missed by other sequencing methods, is a pathogenic mechanism involved in different rare germline disorders. So finally, I cannot finish without giving thanks to all the people involved in our study and all of you for your attention.